This is cells part 4. This is cell transport across membranes. Again, just a reminder about what the membranes look like. We've got our fluid mosaic model. Again, semi-permeable, it lets some things across and keeps some things out. These are the types of movement across a membrane. Diffusion is occurring without any energy. It means that things are going to diffuse from high concentration to low concentration. That is a concept always for diffusion, going from an area of tight space or a lot of stuff to an area of less stuff. Always high to low concentration movement for passive diffusion, which is no energy. I would create a chart like this, so I would pause it here because we're going to talk about each of these things. Facilitated diffusion. If the molecule cannot get across the membrane, it requires a carrier protein. Facilitated diffusion does not require energy. Its driving force is an increase in entropy. They are, again, moving from high to low concentration. Let me see a little image. High concentration on the top, low concentration inside, moving inside the cell. Add to your chart. Active transport. Again, if the membrane cannot get, or the molecules can't get through the membrane, and the molecules are moving against a concentration gradient, which is low to high, the transport protein will be used and energy is needed. This is the cycle of the sodium-potassium channel, which happens in the nervous system. This is active transport because it uses a protein and it requires energy. An electrogenic pump is a pump that pumps ions across the gradient to create a charge gradient. There are proton pumps which move hydrogen ions. There are the sodium-potassium pumps that move sodium-potassium, but an electrogenic pump is a specific type of active transport that moves ions to create charge differences. Add to your chart. Osmosis is the movement of water. Water is, again, going to move from an area of high to low concentration, or it's going to move to the area where there are more solutes or dissolved stuff. So when we look, we see that the water is even, but my concentration of solutes is really high on the right. So water is actually going to move to dissolve that higher concentrated area of solutes. In plant cells, we see this happening. On the left, in a hypotonic solution, this means that there's tons of salt in my cells. Water is going to rush into the cell. It can become lysed or explode, or it can be turgid, which is a normal cell. In an isotonic solution, there's the same amount of salt or a solute in and out of the cell. So water is moving in and out of the cell at the same rate. It will be slightly flaccid. The cell walls will not be kind of tense. In a hypertonic solution, there is more salt outside of the cells, and so water is going to rush outside of the cells. The cell will become plasmalized or shriveled. And you can look up videos of that. You can move that there. Water moving across the membrane. This is passive, doesn't require energy. Uses, obviously, our aquaporins. There are 3 types of endocytosis. It's the movement of larger particles into the cell by the use of a vesicle, phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is cell eating. The larger molecules or particles are brought into the cell by engulfing them and making a plasma membrane vesicle for it. You can look up videos of this. Pinocytosis is cell drinking. It takes dissolved molecules in the extracellular fluid, and it is engulfed with the fluid and brought into the cell in a plasma membrane. And then receptor-mediated endocytosis. There are receptors on the outside of the membrane and allow something to attach to it. When those receptor sites are filled, endocytosis engulfs the whole thing and brings it into the cell. Exocytosis is just the opposite. It's going to release things using the vesicle model again, and releasing it into the outside. You can pause this if you'd like to take a look at it. These concepts should be relatively simple. And that is your chart filled out. Feel free to pause this and copy it down. You need to know this information. This is the blank one if you'd like to see it.